the conversation on metabolic engineering. The title of this conversation is Towards a New Partnership with Nature, Synthetic Biology for a Broken Planet. My name is Michele Catanzaro. I'm a physicist and a science journalist. And I must say, when I was preparing this conversation and having a preparatory chat with the speakers, I heard really mind-boggling stories, ranging from plastic-eating bacteria in the sea or to microbes that produce electricity out of your pee, to things like, for example, bacteria that retain water in the desert, which may have a very interesting impact in our current situation. And in fact, one of the things we will discuss, I think this will be a quite compelling discussion, is whether all this potential can be harnessed somehow to tackle some of the major environmental challenges that we are facing, from climate change to desertification to plastic pollution, etc. But we will also tackle the challenges. So, ethical challenges, economic ones, technological ones, in really harnessing this potential in this new relation with the world of microbes and with nature in general. So the structure of the event will be that there will be a first round of intervention by the speakers. I will introduce them. Then there will be a debate moderated by me among the speakers. And then there will be plenty of time to pose questions from the floors and also from those that are following us from YouTube. So I invite you to take notes if a questions come to your mind, at least a mental note, because we have three very interesting experts, and moreover, three people that are really committed to communication. So there are no stupid questions here. I mean, those questions that are really basic may trigger the best part of the discussion. So I encourage you really to take advantage the people that are here on the floor. So I will introduce them one after the other before they have their first intervention. So there will be some surprise. So first of all, let's start with Víctor de Lorenzo. He is the head of the Laboratory of Environmental Molecular Biology at the National Center for Biotechnology. And one of the topics he has worked is precisely industrial waste, the use of microbes for this specific purpose. And he's thinking hard recently at, let's say, scaling up the solution to global environmental problems. He has plenty of awards, so I will not mention all of them, but they are all very important. And I must say that I think our first interaction was when he co-chaired the President's Science and Technology Council of the European Commission during Durão Barroso administration. So a very interesting position in which he sort of had to advise policymakers, which makes his perspective and his experience especially relevant. So I would like to start, Víctor, by asking you precisely about this. Could you mention a few of the big challenges we are facing and how could microbes play a role in addressing them?
Yeah, of course. Well, first of all, good afternoon, ladies and gentlemen. It's a great pleasure starting this conversation with you today. That is a follow-up of a previous conversation that we had a few months ago on how metabolic engineering and modern synthetic biology can really provide some solutions or some perspectives of solutions to one or some of the major problems that we face as a planet and as a society. So thank you, Michele, for organizing this kind of follow-up. And to answer your questions that I think are very relevant, let me give you a little historical perspective, okay? So I think that the idea that one can use microbes, take them to the laboratory, use the best genetic technology to produce biocatalysts able to degrade important pollutants, started, I would say, in the early 80s. And this interest in this type of developments started also at the time of what you may call the onset of the green awareness in Europe. And you may remember, maybe you are too young, some of you, but that by that time there was the onset and the creation of the Green Party in Germany, and that had a tremendous impact in the whole vision of many societies, in particular Western societies, to bring environmental issues to the political agenda. This is something that had not really happened before. So we knew about disasters here and there, but in fact it was this green awareness that happened in the early and mid 80s that really started to generate some momentum that propagated into the scientific world as well. So by that time, it was also the time of the development of genetic engineering, recombinant DNA technologies, and so on. And, well, a bunch of scientists say, well, why don't we take advantage of this growing public interest in genetic engineering, and together with this possibility and this opportunity to use these new technologies for the sake of improving the environment or to tackle specific problems and so on. We have to say that two major things that happened also during this period of time were, on the one hand, the so-called Exxon Valdez disaster. It was a petroleum tank that collapsed in front of the coasts of Alaska, and then the TVs started to show pictures of, ducks and birds completely covered with petroleum, these type of images that have so much impact in our memory. And at the same time, or by the same time, there was this also big disaster in Bhopal in India, where a chemical factory had a big spill of toxic molecules, and then it was a complete disaster. So this type of things really created a kind of momentum and public opinion that say, well, we have a problem with the environment, and our development as an industry, as a society, as a urban development has consequences. So we better keep an eye on this and try to remediate them. But by that time, also problems were focused in specific places. You had like a petroleum spill in one place, you had an explosion there, you have some local places. And then that had an impact in the type of interest of research in the laboratories of molecular biology and microbiology, in the sense that the focus for a long time was to say, well, we have this site that is polluted with PCBs. So PCBs are chlorinated biphenyls, one of the worst contaminants, and they are very difficult to degrade. They had tremendous consequences in the environment and so on. Okay, so we have this spill there. Let's take a look to what bacteria do to them. So we go to the place, we isolate bacteria that can degrade them, we clone the genes, we study the regulation, we tackle or we kind of modify here and there to have a better process and so on. So for many years, the emphasis was to go to one place, identify one pollutant, and identify one bacterium that was able to degrade the pollutant, take the bacterium to the laboratory, and try to do something with it to improve the process. That lasted, I would say, for about 10 years. But then at the end of the 80s, early 90s and mid-90s, things started to go a bit wrong. Number one, the expectations that were created by the end of the 80s were not really fulfilled. So the public and the funders started to be a bit skeptical about whether, in fact, we could use genetic engineering to make a difference in terms of environmental remediation. But let's face it, there was also the time that public became very sensitive to the use of recombinant DNA technologies, and together with failures and so on. So I would say that by mid of the 90s, the whole field of genetically improving bioremediation, environmental bioremediation, came to a kind of standstill. However, the community that was formerly interested in biodegradation moved massively into this other field of metabolic engineering for the sake of creating industrially interesting and valuable products. So from the scientific point of view, the challenge of degrading something may not be so different of the challenge of creating something. So you go to one place, identify the genes, put a promoter, put this and that, and at the end you create a pathway. And from a technical point of view, the same difficulty is involved in creating a pathway for degradation of something versus creation of something. And obviously, from an economic point of view and from an acceptance point of view, it was far more interesting to have something valuable rather than just destroying something. So I would say that that lasted for a few years, this type of philosophy. Let's focus on using genes that come from the environment, bacteria that come from the environment, to produce valuable compounds. And still to this day, this is a very, very important branch of the whole biotechnological field. However, time passed, and then all of a sudden, we were faced with all this change of a scenario when we faced the data about climate change. And that happened, I don't know, maybe 10, 15 years ago, where massive data on greenhouse gases started to appear, effect on rising temperatures, deterioration of ecosystems. I mean, we started to really face a new situation. So because in that case, the problem was not just to have a big spill of petroleum in Alaska. No, in that case, it was that we were modifying, because of human action, the entire functioning of our planet. And well, that created another type of sensitivity. And then, in my opinion, the time was ripe, perhaps, to go back to the idea of using bacteria, to use microbes, to use advanced molecular biology in the last few years, to use synthetic biology, to really go back to the problem, or to the question, or to the challenge of developing in the laboratory agents that could really have a remediating effect on our misgivings. There's one little comment I want to say in that respect, and it is that there is this kind of benevolent view that we have a problem because of the emissions. So if we stop emissions, everything will be fine. So all we have to do is, instead of having all these tons of CO2 delivered per year, we just decrease it by 50%, and that will be it. Well, ecological theory tells us that this is not completely true, because there's this phenomenon that ecologists and physicists call tipping points. So that means that in some cases, and there's a growing evidence that that happens, ecosystems that are pushed because of our stress beyond of a tipping point will not recover even if the cause of stress has stopped. Okay? So that means that in these cases, it's not enough to just convince the politicians, to convince the industry to do better. No, in this case, we need an active intervention to really change the state of affairs. What are the major problems that perhaps we can tackle with these technologies that we are, new technologies and new way of thinking that I'm just mentioning? Well, I could mention at least 3 or 4. One of them is, of course, the emission of greenhouse gases, not only CO2, but also methane, nitrous oxide, and others. That's a very important issue, and is the one that has perhaps the most important consequences at a global level. But we have at least 2 others that, in my opinion, are super important. One of them is desertification. That means the expansion of dry lands. We see that more and more desertic areas are growing, are expanding, and that's something that is quite difficult to tackle. We have the problem of plastics. That's another big issue that in the last few years has become very, very relevant. And in these 3 cases, CO2 plus other greenhouse gases, desertification, and plastics, molecular biology, synthetic biology, modern biotechnology can make a tremendous difference. The issue is that in this case, we cannot be satisfied with just applying them or delivering them to a small ecosystem. We have to think and to really apply big remedies to big problems. And this phenomenal challenge that we have about climate change asks us to really consider developing the technologies, developing the social mood to go ahead and plan massive interventions in such a way that we can remediate or compensate minimally the type of damage that we have made to our planets. Obviously, this has many different aspects that we probably discuss, but we are claiming here, and at least that's part of my kind of agenda in the last few years, that we should think on large scale, even global scale, planetary scale bioremediation. And the reason why we are not there, in my opinion, is because not enough research has been done in these particular areas. And for this large-scale bioremediation, we need to tackle various challenges. And one big challenge is, of course, to develop the biological agents, the biological pathways, the biological, say, activities that are necessary to convert this phenomenal problem into a challenge that can be tackled with modern science. So in this way, and this is where I want to just finish my little presentation, introductory presentation, I think that this can be one of the tools that we can put on top of the table to heal the damage that we have made to the planet, and perhaps to rethink our interactions with nature, not in terms of domination, but in terms of partnership. Thank you.
Great. Victor, thanks a lot. I must say, as a reporter in science, I really appreciate your historical background, because sometimes in science communication, things seem to appear without, out of the blue, without antecedents, and it's very interesting to see how we have arrived up to here. And thanks also for reminding us that Yeah, I mean that proactive action is needed to tackle issues like, for example, climate change. The second speaker is Rachel Armstrong, a professor of regenerative architecture at QU Leuven in Belgium. She has a degree, a medical degree from the University of Oxford, and a PhD in architecture. So very interesting path in science. She has worked in this concept. She's in fact the leader of the concept of living architecture, an idea, a practice of architecture that is ecological, technological, and life-centered. And she has written several books. The last one is called Safe as Houses, The More Than Human Home. So Rachel, you advocate for sort of different coexistence, let's say, between us and the microbes in the built environment, in our houses, in fact. I was wondering whether you could give us some examples of how this coexistence is actually being deployed in the real world.
Well, thank you for your introduction. Just to follow up from Professor De Lorenzo's observation that bioremediation is an absolute emergency, equally pressing is actually removing the causes of anthropogenic change. And one of the biggest causes of anthropogenic change is the built environment, which currently contributes 40% of the world's carbon footprint. It's about 1% more than transport. So that's pretty significant. And so one of the things that a whole bunch of bioartists and biodesigners have been looking at is how we might change the infrastructure, the systems that underpin how we conduct our daily lives. Because if we think about modernity and the acceleration of anthropogenic change with the rise of the Industrial Revolution and mass manufacturing and mass consumerism and frightful practices for disposing of something that we now call waste. Waste wasn't really a thing until modernity and mass consumerism. So there are deep-rooted expectations about how we live our lives, framed by the spaces that we inhabit. And one of the pioneers of this space for modernity was Le Corbusier, and he described the home as, or the house, as a machine for living in. And if you think about the way that our cities and homes are set up, they're not set up for people. They're set up for machines, okay? And so the question that these artists and designers have been asking, and architects, is what if we change the infrastructure that currently supports machines and actually change that to a life-promoting system? And of course, at the foundations of the world's biosphere are the microbes. So if we think about microbes as a fabric for life that we might want to bring closer into our homes and our cities and organize our daily lives around, maybe we could find a way that we could replace fossil fuels, which is a fuel for the machine, as kind of a supernatural force that comes in a kind of concentrated, geologically kind of potent building block. If we could actually use natural metabolisms within the carrying capacity of the planet, then whilst Professor De Lorenzo is bioremediating all the damage that we've already caused, we are no longer creating the damage for our future generations. So what might a city or a home that could possibly lead us into that space look like? So I'm going to tell you now that this is not entirely fiction. There's a great deal of speculation as we try to figure out what exactly the right moves are. But I firstly want to introduce a number of different technologies that exist today that are, I would say, in prototype or kind of pilot phase of development, or the very first example of their kind. So first of all, in Hamburg, there's the BIQ building, the intelligent building. It was built by Arup and collaborators, that has panels on the outside of the building envelope, that contain algae, little green cows that eat sunlight and carbon dioxide and turn it into biomass. And what these algae are doing is this extra cladding around the building is reducing the thermosolar effect, the heating of buildings passively by sunlight in the summer. And so it's reducing the amount of power that we use in the building to cool it. And so this was a pioneering building that was the first to really incorporate microbes in its infrastructure. The ones that are also quite infrastructural would be things like the P-Power project, which has been running for the last 5 years and longer than that. It's been running as P-Power for 5 years. And it has a pilot that has been present and kind of very much enjoyed in the last 5 years in Glastonbury. And so the P-Power essentially is microbial fuel cells, microbial fuel cell arrays that take the P of festival goers. We could probably do quite a lot with that. But it's turned into bioelectricity that's used to power mobile phones. People can play computer games. And some of that energy will go to power the screens in this pop-up festival. What's interesting is that the group, previously from the University of West of England and now at the University of Southampton, was pioneering that work. I was also working with Oxfam and the Gates Foundation to think of how that microbial fuel cell infrastructure can transform the lives of people without modern infrastructures like sewers and power generating stations. And so they've been looking at refugee camps for displaced people and using the microbial fuel cell toilet. They're able to generate lighting to keep women safe at night around toilets. They clean water, so provide sanitation and provide bioelectricity. So there's a basis of sanitation, cleaned water, not potable, and also a processing system that deals with waste. And these are real infrastructures, and Gates's vision was for communities like this to power around about 30,000 people living in these kinds of temporary settlements. That's a very interesting figure because there's another organisation, a commercial organisation, architects actually, that exist now in the States and in the East and now increasingly in Europe. It's called Organica Inc. They are a group of architects that are transforming human waste and wastewater into food for plants in gardens, and they create greenhouses for urban environments. Again, they can treat water, wastewater, in an urban environment, not in the periphery of the city, between 5 and 30,000 people. So the idea then is that you could get this a kind of resource circularity, taking human waste and cleaning the water through a series of bioprocesses. The root microbiomes of plants are being used to break down the organic matter in different phases. And the thing we would really love to do is to situate microbial fuel cell arrays within this environment so that we could actually produce electricity from this wastewater processing treatment. So in other words, we could literally create power plants. So instead of big chimneys that burn fossil fuels, potentially functionalized gardens like the very one that we're in today, with the right kinds of infrastructure, with the right membranes, the right kinds of systems of flow through these spaces, could produce low power energy. And that is really important, because the microbial fuel cell system could comfortably power around about a 12-volt battery supply, whereas our domestic supplies are eating up 230 volts in Europe. I find that really interesting because we could make a huge difference to our carbon footprint simply by finding ways to develop a 12-volt lifestyle. Not only that, we would be able to be mobile and not depend on city utilities for our electricity, for our water cleaning, and for other functions like providing compost for gardens, or with synthetic biology, maybe even having high-value fractions like bioflavonoids that we could produce at home. So kind of thinking strategically into what a microbially powered infrastructure for homes and cities could be really raises a lot of different kinds of opportunities. And one of the projects that I coordinated and that Barbara and Voltret and Juan were all fantastic collaborators in was a project called the Living Architecture Living Architecture Project, and it was a bioprocessor system that put different kinds of microbial metabolisms together that didn't normally sit together in nature. So we could put anaerobic metabolisms next to photosynthetic metabolisms, and the CSIC made an absolutely mind-blowing, staggeringly interesting, awesome synthetic biology bioprocessor, which I would describe as Charlie and the Chocolate Factory meets a kind of pharmaceuticals outfit. It was a beautiful hack of the microbial fuel cell technology, where light fed these modified algae microorganisms that produced so much sugar that it went through the semi-permeable membrane and it buzzed up these workhorse organisms that the guys at CSIC knew all the genes of, and then they could make new genes, new metabolisms by hacking genes within these different microbes that didn't normally hang out together, but if you gave them enough sugar then they would. And I thought this was just amazing, because now we could bribe microbes into doing stuff that we wanted them to do. And so this leads me to just the final point, which was then, this is cool. This stuff is happening. It's real. But how do we actually interface with this? What does this mean that a home looks like? And so we did a little pilot project off the back of the Living Architecture Project, which was called Alice, which was, how do we talk to our microbes? Because they're not really nice to look at. When we find microbes in our home, we reach for the bleach, right? Because it's kind of slimy and nasty looking and we don't like it. But if we use the electrodes from a microbial fuel cell to let us know how those microbes are feeling, we never have to see them. They can stay in a beautiful ceramic casing like the ones that Voltret makes. And we never have to look at ugly microbes. But we can see how they're doing, because unlike electricity that's used in machinic systems, which is only about power and energy, microbes generate electricity that is also data. It can be used for power, low power, to drive low power electronic systems, but it also is important for chemical change. The transfer of electrons from one atom or molecule to another can actually bring about physical change. And so using microbial electricity, a bioelectricity, gives us a qualitatively new experience of electricity in a home. There's a long history of that. But it also gives us this possibility of an iPad interface where through animations, we know how our microbes are feeling. And so we can change the inputs into our microbial fuel cells or our synthetic consortia. And we can really start to negotiate a kind of microbial economic transaction with them. And I will leave it there.
Thanks, great, thanks a lot. Really, as it had already happened when we had our first conversation, now I really want a house like this one. That is, you have really convinced me. And yeah, I mean, joking apart, it is really fascinating to see that these things are happening, that the potential is not only, something in the future, but it's something that one can see in real world prototypes. And okay, so let me introduce you our third speaker, Ricard Solé. He's a physicist and a biologist, and he's an ICREA research professor at Pompeu Fabra University in Barcelona, where he leads the complex system labs. He has focused much of his recent research in the evolutionary origins of complexity, and he is the author of the concept of liquid brains, which is a framework to develop a theory of cognitive networks. Another concept that he's working on a lot recently, and that I think will be very relevant in this context, is the idea of biosphere terraformation, that is, engineering ecosystem to restore degraded habitats or avoid tipping points. So, Ricard, I would like to start precisely from this last point. When we had our conversation, you mentioned that you are planning to kick off a project to check whether modified microbes could trigger a biosphere in an especially challenging context, which is arid ecosystems. So, and even one could imagine in something even more challenging, which is the outer space, another planet. So I was wondering whether you could tell us something about this project.
Okay. Well, you all know that Elon Musk said that in 10 years we'll go to Mars. So it's no reason to worry about everything. Right? But yeah, when Victor was talking, it came to my mind. I wanted to kind of also make a historical picture of this. I remember 30 years ago, something like that, somebody came to the Santa Fe Institute, where I also worked part of the year, and he was making a kind of a forecast of climate change in the future, etc., etc. And you know, when you live enough to see what things happen, I remember well when he made these predictions that seemed kind of very specific, like huge floods in Central Europe, huge mega fires in Australia and North America, and so on and so forth. Plus all the story of what's going to happen with when we get run off of fools, and we have a peak oil, and you know, just saying that it's not that like scientists have not been able to predict what's going to happen. And actually, sadly, what we see nowadays is that the things that were not well predicted is that because they are worse than we thought. And I just don't want to be so pessimistic, but it's good to have an idea of why we're talking about climatic emergency. And I don't think it's because of chance that politicians now start to kind of seem to be worried, right? Because until a few years ago, it was like, we'll do something someday. And so the idea of terraformation, I wanted to make the point of Mars because we borrow the word from there. But of course, it's not like terraforming a planet to make it more human-like or good for humans. And I just want to make the point that Mars is there to kill us. I mean, it's not a place to live, right? But from different sources of inspiration, I've been working on theoretical ecology for many years. And at some point, we started to work in synthetic biology. We have our own wet lab, and we realized all the problems that real experiments imply. And from a number of influences came the idea of why not to consider the idea of terraforming our planet and our ecosystems. And I try to make the point of why is this something that is sensible and why is reasonable, right? And I want to say that one of the inspirations I had was a short article by Rachel that she published in Nature called, I think it was Living Quarters, where she made this very nice, you could say speculation, but I think it was quite reasonable, that what about attacking the problem of pollution in a place like the Venice Laguna, like building an artificial reef and colonizing the reef with, you know, microorganisms that you might have actually designed. So that's what it makes sense. And the first thing that comes to your mind, of course, is the Jurassic Park picture, right? So you manipulate something, you bring it in nature, and so what's going to happen? What's the unintended consequences, right? Well, this is a completely wrong picture for many reasons, and I want to also make the point clear. First of all, because it's not something that we can solve scientifically, right? If I ask you, do you believe that there's life in other parts of the universe? 99% of people will say yes. But if we are honest, why you say that? We don't have evidence for that, right? So why do you think there's going to be unintended consequences if we deliver something that has been manipulated genetically, right? And we can talk about that later, about the wrong analogies with invader species. That's not the idea at all. And also because, I mean, Victor briefly mentioned that in the 80s there was, this excitement, but also came the problem of banning any kind of research concerning GMOs to be delivered in nature. Not because there was any evidence that that could be harmful in any way, just because, something totally non-scientific happened, and so that some companies that use GMOs have behaved in a quiet, non-moral way didn't help, right, over the last decades. But the point is, this has not been tested, has to be tested. But let me tell you some things. We have been working on that actively in the last 6 years, doing a lot of mathematical and computational modeling, which is kind of the basis you do in ecology when you want to try to understand a system. And the interesting thing is that you can actually show that, for example, if you want to do something that is absolutely essential, it's not usually in this narrative. But if you design something that protects biodiversity is something essential to maintain the quality of ecosystems and make them resilient. So you can design modified organisms to put them in ecosystems. We have a conjecture on that, and preserve biodiversity and make them be functional. The example of arid ecosystems has been one of the main threads of our work, and the reason is there are several reasons for that. Although you usually think in the Amazonian rainforest and the more kind of iconic systems, arid ecosystems, drylands, is about almost 40% of land ecosystems, and almost a third of the world population lives there. Right? It's not like something that is external to us. It's very, very important, and they are going to experience shifts in the future. That was briefly mentioned by Michele, that things are not going to happen smoothly, and which is something that's essential for understanding the risks and planning for the risks. Because if we believe in a linear world where things are happening continuously, we can always think that, well, someday when things go really wrong, we'll go back, right? And that doesn't work. Complex systems do not work like that way, right? There are tipping points. There are points of no return. That happened in the past, and we see them happening now. Although we need to know more about how to monitor that and how to understand that. Choosing arid ecosystems is very clear why. I mean, these systems, we have quite good knowledge about their ecology, in particular of the soils, and in particular of some of the key species that play a really leading role. The point here is, and that's our proposal. Why about taking something that is already there? That's an essential part of the story. Something that is already in the community that has co-evolved with the biodiversity that you have there. You take it, right? Our conjecture is a cyanobacteria. You make a little modification, like it will secrete a molecule that retains a little more moisture or improves the quality of the soil. And that has two implications. One is that, that's our prediction that we have to test, that biodiversity is kept. So these microorganisms will not propagate anything into the system, and that a little difference like this in real systems that are highly nonlinear can make the tipping point be pushed away, right? You can even maybe transform the nature of the transition, right? And if we need something now, it's time. That's pretty much fundamental, and that will have great implications. Arid ecosystems, we have a lot of indirect evidence that this kind of strategy could work. And we've been fighting a lot to get money, that's part of the sport in the scientists, is using time for writing grants. And I actually remember the first grant we wrote for Sinterra, and Víctor and Rachel were there involved. And I remember also a picture we have with many different case studies. It was from Venice to the biosphere, and I think it was too ambitious maybe. But now we have an opportunity. There's a joint effort with EMBL, the European Molecular Biology Lab, who has made a strong bet for working on ecosystem level and global issues on biodiversity. And so try for the first time to build a microcosms, because we need to start here, right? To try in the lab with control conditions, realistic conditions, to see if we can actually do that kind of intervention. And that opens a lot of opportunities for doing research. And just wanted to mention that one of the most interesting things of this area, beyond the obvious implications, is that it will bring new science. Mm-hmm. Because it's easy to find a synthetic biologist who can tell you about genetic constructs and all kinds of designs and metabolic engineering at the microbial level. And you have then the community ecologists, the people who've been working for 50 years in understanding communities, and they can tell you about invasion ecology, community organization, resilience, and essentially the two extremes of this very large array of scales is not yet connected. Mm-hmm. And there's a lot of things that we have to learn from each other.
Great, thanks a lot, Ricard, for bringing a lot of nuance to this concept. I mean, I think it's really interesting, this macro and micro approach, you know, from the gene to the whole structure of the ecosystem. And also, I think it's fascinating, this idea of, you know, taking something that is already there and, you know, using the potential of something that is already integrated in the ecological network. So now we move to the debate. We have around a little less than 20 minutes for the debate, and then I will open to the floor. Ricard, if you don't mind, could you give me one of those mics? Because since we have only 3 mics, I will try. Does it work? Okay, I will try to use this one if it doesn't interfere, so that everybody on the table is free to speak whenever they want. So let me first very quickly make, let's say, one step back. Now we have talked about, you know, really futuristic applications, you know, front line. But I mean, we have lived together with microbes. We live together with microbes, you know, since forever. Okay, and we have also used them a lot in traditional ways. So I would like to ask you, to whoever in the table wants to comment about this fact, about this idea that, you know, microbes, bacteria, we have associate them with something that is bad, that is dirty, that we need to fight, but then instead there are plenty of situations in which they are not bad at all, both in nature, in our body, and in technology, in very, you know, technology that we deal with every day. So whoever wants to comment, maybe Victor, if you want to start.
So, Michele, you made a good point that for the general public, bacteria are bad. That's the bottom line. And, well, in reality, we all know that, well, we all know, there's a growing evidence that we are in a microbial planet, basically a bacterial planet, a microbial planet. So bacteria were there much before we arrived, and we remain here much after we go. So in that respect, bacteria are the major contributors to the Earth biomass. I want to make a little parenthesis here. So there has been a recent calculation of how much biomass is in the biosphere and who are the actors that contribute most. And in full, say, weight, plants are the major contributors, but if we exclude just one molecule, like cellulose or lignocellulose, and then we delete that from the equation, then the major contributors of the entire biomass of the biosphere are bacteria. So we basically are in a bacterial planet or a microbial planet. In that respect, the history of Earth have shown that bacteria have been the prime agents for major climatic changes, not only one, but various times. Episodes that we have a geological record of big climate changes in planet Earth have been, for the most part, triggered by bacteria or by microbes in general. And that means that bacteria and microbes in general are our main, if not our only allies to make a difference in terms of climatic management. And this is something that is good news, in my opinion, because we have this growing, say, technology that allows us to manipulate, or, well, manipulate sounds like negative, allow us to program or to reprogram or to start a kind of conversation with the microbiome to change its behavior. And if we are able to really convince the microbiome to deal with our emissions differently, probably that will have tremendous consequences for the whole health of our planet. And I insist that, perhaps with this idea of a new partnership, a new covenant with the environment, we should bring in the microbial world as our main, partner in this conversation.
And they are inside us. I mean, about 2 kilograms of our body is estimated to be. Do you have any additional remark on this?
Yeah, I'll take it from an anthropocentric perspective, because effectively we demonized microbes during the reign of hygiene. And in the late 19th century, there were two scientific realms that were being discovered. One was the relationship between microbes and clinical health, or the health of the human body, and the other one was environmental. But because 1/3 of all adults were dying from infectious disease, from plague to cholera, that the relationship between the kind of pathogens took the spotlight. And so politically and society mobilized against microbes and went to war with them, which is why we then sought out things like antibiotics. And I think that Professor De Lorenzo has something to say about our use of war metaphors. But it set the scene for modernity with this idea of the reign of hygiene, where all microbes were bad, and then we developed domestic products, which we then tipped down our kitchen sinks and into our rivers, which then led to Rachel Carson's Silent Spring. So it is our demonization of the less than 1% of all microbes that do have pathogenicity that has led to this great kind of twisted view of what a microbe is and what it does.
Yeah, I will add that one of the great things of the understanding our own microbiome, and particularly the gut microbiome, has brought a lot of interesting insight for the Terraformation project, because you of course could bring the question of if you mess with the biosphere or ecosystems, we have only one biosphere. So any other test. And the good news is that now it's a huge amount of work that's been done in biomedical research on actually bringing new microorganisms into the gut to actually correct diseases. And you can actually, thanks to omic techniques, you can actually follow up what's the impact of that. And that's giving us a lot of the real insight, because the microbiome is nothing but an ecosystem, a really complex one with a lot of similarities with a real ecosystem like the rainforest. And so that's our parallel experiments.
Mm-hmm. Yeah, and also let me remind you that somebody was having a microbial produced product, a beer here, and also cheese and wine and all these sort of things, or even compost, waste processing, etc. There are plenty of ways in which microbes are already being used, in fact, in a very mainstream way. And I mean, when we talk about metabolic engineering, synthetic biology, one other, I think, background point that one should bear in mind is that microbes in nature are quite promiscuous in terms of their genes, okay? So they have, maybe, could anybody want to comment on this part, on the fact that they really interchange genes quite normally, let's say, independently of the fact that we touch them or not?
Well, that's one of the important points we used to make when discuss this question of the unintended consequences. Because if you're talking about modifying a single gene from a given microorganism, it might sound like a lot already, but in fact, as you are saying, there's this horizontal gene transfer. There's a constant messing of genes around, and I mean, and it's nothing like Frankenstein coming out from anywhere. It's really part of the way of the resilience of nature, with the mixing of the genetic and the cellular levels, and adaptation coming out from that.
So to be clear, bacteria do not only inherit, let's say, their genes, but they interchange genes horizontally throughout their life, let's say, okay?
Yeah, well, as a matter of fact, this is, again, sometimes considered as a problem because of the spread of antibiotic resistances that happen through horizontal gene transfer of bacteria who have genes for resistance of this particular antibiotic to the others and so. But on the other hand, I would say that horizontal gene transfer is like kind of internet of microbial communities, the way that they transfer information here and there. And as a matter of fact, in my laboratory and other laboratories, we think this as a phenomenon that could be leveraged to really spread information, beneficial information from an environmental point of view through a wider bacterial or microbial community. So the idea that we are entertaining, and others are along the same line, is that perhaps if you want to enter a new biological activity in a pre-established ecosystem, rather than bringing a new member, engineer or not, into the action theater, you may want perhaps to spread in the existing community a new information, a new activity. And in my opinion, this can be one, say, big breakthrough in terms of biotechnological and bioremediation technologies. Not so much to enter or to release or to kind of spread out specific strains, but to spread the information. And this is something that we are actively working on.
Yeah, and so...
I was just going to make a non-scientific perspective, which is like both Ricard and Professor De Lorenzo have noted that actually the majority of creatures on this planet are not humans, and so it's really twisted that we get this Noah's Ark like notion of vertical descent as being the proper way of conducting ourselves and call microbes promiscuous. I think really the question is, why don't we do that? Maybe we would be with our environments in much better ways if we adopted some of these kinds of more experimental ways of living. I mean, our trade-off is that we develop form, and we like that. But I just think that we sometimes need to invert these questions.
Yes, sorry for being moralistic with microbes. But yeah, so now that we have sort of set the stage, I would like to ask a basic question. I mean, maybe most of people here already know it, but could you briefly, Victor, explain what is metabolic engineering, what is synthetic biology? This is an important point for somebody that may be following us on Twitter. And so very briefly, I mean, just to...
So one of the challenges in biology is that unlike chemistry and unlike physics, in biology we resort to metaphors all the time, okay? So metabolic engineering is a kind of metaphor in the sense that what we try, or the community involved in this endeavor tries, is to rationally design metabolic properties in bacteria or other biological systems by rational combination of specific genes that encode the specific activities together with control elements. Okay, so in nature, for a pathway to work, you need a number of genes, enzyme A, enzyme B, enzyme C, that is controlled by some regulatory signals in the DNA that are called promoters or Schneider-Garner sequences. I mean, this is the jargon of molecular biologists, and this is the way nature does things. So what we call metabolic engineers say, hey, nature has done this in this way, but maybe we can do it differently, and we can use the bits and parts that nature gives to us, but put them differently to create a new thing. It's like if you have a Meccano, you know, or a Lego, you know, and you have the bricks here and there, and with the same bricks that are given by default from nature, then you can reconstruct them following a rational, say, blueprint. And at the end, what we expect, and in many cases it has been very successful, is to have biological materials, in many cases microorganisms, able to do things that nature is unable to do. It's what we call new to nature properties. And well, I think that in a couple of words, this is what metabolic engineering is about.
Thanks. I wanted to ask Rachel, I mean, you are an expert in design and design thinking. I was wondering how this idea of interacting more with microbes changes the way we design things. I mean, how it changes, let's say, the standard flow of thinking.
Yeah, well, I think once you have a substrate for design that's living, it has an agency. It wants to do stuff all by itself without you. And so essentially you're not really in a situation of complete control. You're setting up a negotiation. And how you go about that, I think, is really critical to what the outcomes are. What fascinates me about synthetic biology and the idea that we can engineer microbes is there is a kind of an intention for control and driving outcomes, when I think there's actually possibly more of a diplomatic environment that's needed because of the different ways that microbes themselves read and respond to their environment. And I think synthetic biology is helping us learn some of the communications tools that we need in order to have what actually in critical theories are called a parliament of things. So a parliament of things is an idea that's been developed by people like Isabel Stengers and Bruno Latour, which says that in a kind of a natural environment, agents negotiate stuff on their own terms. And microbes are really good at not just dealing with their own species. They've got, Bonnie Bassler says that there are kind of different ways that microbes have conversations, and they not only just talk to their mates, they can talk to, not even just different races or cultures, but they can talk to other species. And so that degree of communication through persuasion and intervention and this kind of negotiated marketplace of transactions, I think is really, really interesting. Because it's no longer about command and control and what degrees of control we can inflict upon microbes, but how can we persuade them to do the kinds of stuff that we think are fun. And for me, kind of entering then into a design environment, it kind of, it asks different voices to the table when we're thinking about some of these shared challenges that we have, and maybe different perspectives and levels at which we may engage. So that's essentially the fundamental issues. When you've got living things, you're no longer boss.
Yeah, I think somebody else in the table used the same word of persuading microbes to do things, so it seems that there is a common thinking behind this. And Ricard, you have mentioned this nuance of the importance of, taking something that is already there and using it. But at the same time, I mean, the vast majority of the microbial world is unknown to us, okay? So I was wondering whether the strategy may be another one, which may be scouting for microbes that do the things you want, okay? And I would like to hear your thoughts and whoever else wants.
Yeah, absolutely. I mean, one of the reasons we in a way, decide to push forward the idea of using synthetic biology is that we can keep the message from community ecology, having in mind the network of species, so have the ecology at work. And at the same time, taking something that is in the system, and the reason of that is that, as I was saying, biodiversity protects the system from invaders. And since we can make a specific intervention, that I know is not always as simple as that, but in principle, we can pretty much get well informed about what exactly has happened, and then read the consequences of that. But that doesn't mean that we need to necessarily do that. Once we were able to identify the real advantages of doing some particular interventions, we might actually use all the resources and big databases that are there to actually identify potential candidates that can more or less adjust to what we want. Because we also know that ecosystems have some degree of redundancy and capacity for adapting with new strains that come without being invaders. So yeah, the door is open for all that kind of technology.
Mm-hmm. Okay. So one last question, maybe before we open to the floor, is precisely dealing with the challenges. Okay, so this has come up in the conversation at many points. That of course the idea of take an organism, modify it, release it, etc. That immediately sounds like hey, stop! What are you doing? Okay, so Ricard has already mentioned something about it. Yeah, I mean, could you talk about under which condition these can be done basically safely and it's not a problem, and whether there are some caveats or some way of doing things that one can consider as the serious way of doing things and the not serious one, let's say. Any comment of how to deal with this issue?
Yeah, well, there's a kind of historical answer to your question as well. So it's really paradoxical that the traditional concern of big sectors of the public is what happens when you put a new microbe in the environment. It will destroy the community, it will propagate like crazy, it will spread this and that. Well, for us, interested in bioremediation and that would like to put things into the environment for degradation of this or that, the challenge is just the opposite, namely how to put something into the environment that survives and works. Mm-hmm. Because, what happens with bacteria is a bit what happens with domesticated animals. You bring into the laboratory, they get used to the good life of the laboratory, and then once you put them back into the environment, they die, and they disappear very quickly. there's one interesting thing, you know that there have been many surveys of metagenomic DNA all over the world. All these expeditions to collect samples from all types of places, and you have thousands and thousands of genomes here and there and there. To the best of my knowledge, not a single hint of recombinant DNA has been found in these databases, okay? That means that, I'm sure that the recombinant DNA has been released accidentally thousands of times, mostly at the beginning of the recombinant DNA era, where people just, throw the cultures to the sink with no care and so on. These genes and the sequences have virtually disappeared. So that, to me, is an indication that, for the time being, following what Ricard said, we don't have any evidence that we have this danger. And in my opinion, the problem or the challenge is just the opposite. If we produce something interesting in the laboratory, how we make sure that either the genes or the agent itself is able to propagate and increase the beneficial activity for environment that we have engineered in the laboratory.
Mm-hmm.
Can I follow up with a non-scientific observation, which is, I think this is still a function of the them and us scenario, that we are at war with microbes and that we've got to be boss. And we haven't positioned ourselves in a microbial marketplace as human traders yet. And I think that there is an ethics and a certain kind of view of what we're engaged in, that really, we really need to change the language and we need to change the conversation because we're so happy with our war metaphors and kind of creating these conflicts rather than setting up situations where there's a kind of diplomacy at work, a kind of, let's say, a benevolent surveillance, a being aware of and taking responsibility for and thinking through the consequences of our actions. We've become very comfortable not doing that during modernity. With machine, you can press a button and you expect all the problems to go away. That's been our expectation. When we're in a living world, we can't do that. We have to stay engaged. And so that's why I think it's really important that the conversations around this issue actually start to change, and that the fact that we're hacking genes is actually possibly a sign of our primitive understanding of who microbes are, and we need to get them to know them a lot better.
Yeah, I would like to make a final point that I think that helps in a way to put in the right context the proposed idea of terraforming with other alternatives. And I use that because I think that usually makes clear why the questions and the way we pose the questions are wrong. Once you explain this idea that I want to use synthetic biology to create an organism, the liver, everybody gets concerned. But then, if you explain what was done in China, for example, more than 50 years, when they had this big travel in an area which was semi-arid and then arid, and then sand started to cover the railroads, they decided to do something about it. And what they did, this is Chinese scale, was to do the following. Now, but this is an amazing stuff. They used to do, well, we'll do a big intervention. So we'll bring thousands of tons of haystack and dried grass from somewhere to nearby the railroad. So they actually put all there. You have to see the pictures because it's kind of I think it was a half a kilometer by 9 kilometers square kind of shape, kind of chessboard. And they kept making interventions, like them bringing shrubs and then, So who is with me? You're saying you put a one gene in one species that was there and that could be a problem. But what about the thousands of species, exotic, that you're bringing from somewhere else to this particular place, right? Because this is surrounded by real ecosystems. So we need to put the questions in the right context, because it is no sense to say that one intervention is okay, it's nothing else going to happen, and the other is necessarily going to create big damage.
Yeah. Okay, so now it's time for an open discussion, and I must say I was in the previous debate in this series, and one thing I really appreciated is that it was a two-way discussion. So, for example, some ideas came from the public that we took into account in preparing this debate, and I really think, I encourage you to make your comments, remarks, etc. So in order to start the debate, we have a row zero. I will introduce the people that will trigger the debate. So first of all, Neptune Yousefi. She's a PhD student at the University of Vienna in the Polymer and Engineering group. And she works in adding carbon nanotubes or graphene into composite matrix. And she has also worked with bacteria, in fact, in a project called co-corporeality. And then Monica Brandich, she's a PhD researcher in the Hub of Biotechnology in the built environment at Newcastle University, and she also works at NASA Ames Research Center on biotechnology solutions for building habitable environment in space. So please start with your comments or question or remarks.
Hello. Okay. Because I'm a short person, I have to stand up.
Oh, no.
That's not it.
Okay, thank you so much for your lovely input and everything. So, actually I need my notes. So I have a lot of questions, but I'm going to reduce it to a few for the beginning. So I was interested in this modifying of bacteria. I was wondering how much time does it take? And also it seems like you need the material or the molecule you're looking at to be degradable. Otherwise it's not working. And I'm addressing it too.
Okay, well, genetic technologies in the last few years have really grown exponentially. So now basically we can program entire genomes with the degree of reliability that a programmer can program a computer. I mean, right now, the question is not about cloning, it's not about, mostly it's about writing the right DNA sequences. And then, in the future, in the near future, that will be as easy as when you put up new software in a machine. So in that respect, this programming is backed by an immense number of platforms that will assist you to basically do nearly whatever you want. So just to give you an example, so there are various computational platforms that say, well, I want to create this molecule, and you paint with in the computer the shape of the molecule. How can I do it? You press a button, and then immediately you have the list of genes that you require, the sequences of the genes, where you can get them, and how they could be put together in case that you want to use them. On top of that, the system will give you a thermodynamical and kinetic prediction on how the system will work. And even before having the thing in your hands, you have an idea of whether your endeavor will work or not. So your question and your concern at this point is virtually solved. That's not a problem any longer. And the limitations are somewhere else.
So are we talking about the years, or is it?
No, I mean, years, I mean, the process may take seconds, from, identifying the molecule that you want to design, at least in silico, the pathway that you need to deliver that compound. Then you make the experiments, they may not work, then you have to go into this engineering cycle of testing and and error and all these things. But the first prototype can be obtained in seconds, so that's why, I mean, the technology is skyrocketing at an amazing pace, to the point that people say that in the near future, we will rely on artificial intelligence because we really cannot even handle, the pace of generation of information on these things, right?
So my second question would be about heavy metal ions. Do you think there are bacteria out there that are capable of capturing those, because they're quite a big problem?
Yeah, well, I'm glad that you asked this question because, we have been quite interested in my laboratory on that. We had some works that were made time ago on engineering soil bacteria. For instance, there is a bacteria called Rastonia, that is a soil inhabitant, where we engineer, for instance, on the surface of Rastonia, metallothioneins. Metallothioneins are proteins that bind metals with high affinity, and they produce in the liver of mammals and so on. So we engineer bacteria that express these proteins and put them in soil, and we could see that by introducing these bacteria in soil, then we could recover the vegetal cover in places that were contaminated with cadmium, for instance. This is just one example, okay? There are other examples. And, well, indeed, the problem with metals is that they cannot be ultimately degraded. So they can be blocked, or they can be the chemical species can be made less toxic. But as you know, I mean, either you contain them or you cannot get rid of them. Yeah, but there are ways to handle that, not to solve the problem, but at least to manage the problem.
Maybe let's hear Monica and whether and let me remind the people that are following us from YouTube that I'm checking the comment box. So if you have questions, I will ask them for you. But
okay, thank you so much for this fascinating discussion. I really like where my brain was going while listening to you, because it was so many different subjects, but also connected with each other. I don't really have questions per se. I have some thoughts that I hope will trigger more comments and more discussion. So from what we were discussing, I had this this quote on my mind by William Gibson, who said that the future is already here. It's just not very evenly distributed, in the sense that we already have solutions for so many different things and ideas and technologies and the ideas how to use biology and bioengineering. But now it's up to us how we will shape it all together. So we have this power of maybe not predicting the future, but proposing solutions. And that brought me to what you were talking about in the climate change and environmental challenges, where it's the point where we cannot really be only responding to these challenges, but we have to be proactive in a response to environmental challenges, and also like how we change our living here on Earth. And that brought me to Mars, as Ricard was already introducing, and that's also the field that I'm working with, so I had the most comments about space exploration and space architecture, where in the contrary to Earth, where we're responding to challenges, and with microbiology and microengineering, we're trying to redesign the way of thinking about how we can solve architecture, construction, and in general our living. In space, it's more that we are designing it from scratch. So microbiology is becoming the enabling technologies for that. So I think that there's this beautiful connection, and people very often questioning why we go to space where we have so many problems here on Earth. But when it comes to microbiology and biotechnologies in general, I really see this link where in space we cannot really do things the way that they're done here on Earth, because it's completely different environment. So we're really pushed to think differently, and that change of perspective can be really brought back here on Earth, where on Earth we can take shortcuts and somehow not solve everything the way we at first imagine, but in space you cannot really do it. You have to solve it like completely, and then it can be brought back here on Earth. So yeah.
Thanks a lot. Yeah. Any thought on especially this point of how all the space thing could help us?
Yeah, I mean, we have a paper. I mean, I don't want to make me be an anti-Mars colonization thing person. I mean, it just was criticizing Elon Musk in particular. But I do hope there's one day that a mission goes there, right? Because that would be fascinating. But we actually have a paper where we compare Mars, the microbiome, and drylands, because there's a number of common points that we try to explore, from metabolism to a number of universal features, and conjecture what kind of microbial consortium could actually succeed, right? And we ended up thinking about hypercycles and things like that. But I do believe, and it's part of our research on synthetic ecosystems, the closed ecosystems, because indeed there's a challenge that has been there since Biosphere 2, and a lot of really interesting questions. And one of them, for example, that we have been recently working on, it's about soil itself, I mean the material, because it's becoming more and more clear that you cannot look at the soil on Earth at least, right? Like a kind of inert matrix where bacteria grow is actually part of an extended phenotype. It's quite an amazing story that you can actually build, and you rethink a lot of ideas. And that I hope can extrapolate into thinking of the big problem, I guess, which is what to do with the soil in Mars to make it profitable in different ways, both for the applicability and for the basic science. Yeah, I don't know.
I think I'll just make a short comment, which is, Buckminster Fuller is one of the people credited with the notion of the best way of predicting the future is to design it. And I think one of the people who's really kind of pushed out thinking in that space is Michael Mortner, who's an astrochemist. And he's written a really beautiful book about panspermia. But what's interesting about it is he has this theory of kind of horizontal relationships between people and microbes. And I guess the point I want to make about that is, so he's saying that actually people aren't the best creatures to go and colonize in uninhabitable, desolate kind of landscapes, you know, the other side of the universe. But microbes might be, and that he proposes that there's like a community of DNA, so that we all know that we're terrestrial, but we've kind of got all these different forms. So he's got this kind of theory about a kind of an ecology of settlement. And I think the thing that I would suggest is that we need to de-anthropocentrize these ambitions, because we still, you know, the world and the universe still revolves around us. And I think when we don't do that, I think we'll see a lot more possibilities for better design in which we can realize those futures that we need.
So once again, ecological thinking as an important lead to think about these topics. Okay, so before maybe going back to you with more questions and comments, let me hear whether there are any other questions, comments, remarks in the floor, please.
Yeah, first, thanks for being here and for the discussion. I'm Tobias Goes from the German Institute of Human Nutrition, microbiologist by myself, and I want to give a little bit of contra to your comment on your question. It sounded like it's super simple. Yeah, press one button, second, everything is here. Of course, it's not the case. I want to remark, the first step is always relatively easy. Yeah, you have a thought, you work on your computer, maybe one day, maybe 1 second, maybe a week later you have some results. Then you go into the laboratory and you try first trials, maybe take 1 month, maybe take a year. Then you scale a little bit up, either degradation or biosynthesis, whatever, takes maybe 2 years. But then the biggest step is, I guess you know that, going into practical use. I worked on biodegradation of chlorinated phenols and stuff like that. We are just at the beginning. Yeah? We know these things for 30 years, they happen. But last year or the year before, it was the first practical use, so that you get an impression what was, what is, and maybe in the future, I hope so, from theory to practical use, it will be faster. But it's definitely not the way you go press a button, second, next year you have a practical use. That's not the case. Also, for example, for regenerative energy, I was involved in a project on hydrogen formation by microbes. The first wave came into the 80s, as you correctly said, and that lasted for 10 years. Then people saw money's gone, no hydrogen is produced. Second wave came in 2005 to 2015, again the same thing. I hope the third wave or fourth wave will be better, but indeed we are at the beginning. I think we are in a transition phase to a more microbial society, and I hope that, because I'm really impressed what microbes can do, not only in theory, but what they are already doing. On both sides, microbes produce methane. It's a climate gas. They take up CO2, they produce CO2, whatever. They are capable basically of everything, especially with metabolic engineering. So, just to give another perspective.
Yeah, thanks a lot for this remark.
Yeah, well,
Do you want to comment? Yes.
well, I want to qualify what I said before. Making a prototype in silico is easy, but as Boris said very rightly, then you have to do it, and then it doesn't work, and you have to go again, and you have to try this, and you have to try. So making the prototype is easy. Then going to the practical application takes time. Okay, but even making the prototype that in the past was a pain in the neck, right now is very, very easy. So that and this distance between predictions and applications that in the past have taken so long, the times are shortening and shortening and shortening, and maybe in the not so distant future we'll see this time being very, very short.
Any other question, comment, remark, please? Raise your hand so that I can...
Hi.
Thanks.
Amigos. So I really liked actually your work and your work independently. And I like
Not mine, so...
Sorry, I'm sorry.
No, I understand, don't worry.
And yeah, maybe you're a piece of the answer to this question. But and I like even more that you are in connection and that you somehow started talking about this and now you are together in this table. So I would say, I know my question was, what are the challenges for translating the outcomes of interdisciplinary conversations into progress? And I think, well, maybe one of the answers is
Communication.
communication.
Okay. But let's hear the real experts.
Yeah, and also, and the second question would be, in the case that there is some sort of oscillatory opportunities in history to do such a change, where are we now in that oscillatory situation?
In a peak or in a very...
Yeah, well, in relation to your second issue, so I want to share with you a concern that I have, and is about the fact that many branches of biotechnology have an instant, what you may call, market demand. So that means you put a new drug in the market for some therapy, or you put a new product, or immediately you have a market for it. In the case of the technologies that we are discussing, reality is that no one will make big money with them directly. So that means that the type of market frame for this type of developments is completely different from other types of biotechnology. So, you say, how we can accelerate these developments? Well, the system will be super accelerated if someone finds a way to convert our technologies in making money. And that's tragic, in my opinion. But for the time being, this is what is happening. Mm-hmm. So bioremediation right now, that has been like, under the carpet for a while, all of a sudden, Biden made a claim that he's going to put this many billions in fixing the environment. It was before the war in Ukraine. Like in weeks, you could see a large number of spin-offs in the US claiming, applying bioremediation with synthetic biology tools and so on, that were unheard of before Biden made this bid. So unfortunately, and this is something that worries me, in our kind of socio-economical frame, unless someone makes money, things will not happen, and that's tragic, and I don't know how to get away with that.
So it's a very different scenario with respect to GMOs, for
Absolutely,
example, in which there were big corporations interested in this place. It's the other way around. Any other remark on either these questions or the other one, that is how to make interdisciplinarity and complex conversation between different stakeholders become real-world progress?
Oh, hell yeah. I think that Professor Lorenzo's comment is absolutely right on the money. We need different so interdisciplinarity, I mean, people have been doing that before the Enlightenment. I mean, there was the trivium and quadrivium for education before we kind of siloed people out into kind of monodisciplines. The issue is the paralysis of the marketplace, the neoliberalism and the market deciding everything. We need politicians who are prepared to take value-based decisions and support the development of science and knowledge to the point where other kinds of, so I think we need to look at microeconomies. In other words, they're very locally produced. So you were talking about, the kind of the heterogeneity, the unevenly distributed notion of the future. Well, that's how nature is. And I think that there are more microeconomies based on local resource availability, local crafts, local ways of living, because it's nice weather or it's terrible weather. And I think there's more diversity in economic systems that is needed, so that we're not all trying to be a super rich billionaire that then can kind of decide whether he's going to spend his pocket money on a new widget. That's absolutely dysfunctional. So I think one of the things that we really need is a commons, and a commons of innovation. And what I mean by that is, at the moment, all scientific knowledge kind of goes towards becoming a little company, and then it becomes a big company, and then it becomes a multinational corporation, then it pushes up the GDP, and we learn how to consume. You know, the kind of capitalist society, when it's only capitalism, deals with deprivation and excess, and that's not a fair way of doing things. It doesn't mean that there's no value in it at all. But if it's the only system that we engage in, then you end up with unacceptable inequalities. So if we had microeconomies where people can put things away from themselves, but they never become a billionaire, and if we had commons where there is actually more active sharing, private ownership goes down, individual profits go down, but there is a kind of collective resource, a bit like the term for the microbial commons. Microbes actually have a commons of goods that are divvied out with different rules. I'm not the expert in this, but the sharing systems and common ownership, including the sharing of ideas. Before the Enlightenment, we didn't try to IP our thoughts. When we told a story, we knew we were telling a story that's probably been told before, but we did it our way and we shared it. So this idea of ownership, I think, is really creating distortions in the way we work together. It's making we've valorized competition, and then we say, interdisciplinarity is a little bit collaboration. Everybody goes and competes in their corners again. So there's some really fundamental shifts that need to happen. Bête noire.
Yeah, I will add about interdisciplinarity. I think that for this particular area, it's essential that, as I was saying before, we bring together people from ecology and people working on molecular biology, metabolomics, etc. For a number of reasons. But for example, just to illustrate the point, there's been a lot of effort precisely with this motto of what are the unintended consequences of design principles and in designing genetic kill circuits for you have a bacterium, it does some function, and then to prevent that something is going to happen, you kill it. And the reason of that, to some extent, is we still are using the metaphor of invaders as the metaphor for modified organisms. And it's very easy in discussions like this, somebody brings the idea, what happened with the rabbits in Australia, right? Right, the rabbits, you brought the rabbits to Australia, they spread all over the place, and they made a major catastrophe. But someone should mention that before them that, they killed every single predator. They brought, 10 million sheep. Plus, you probably don't know that, a lot of camels, right? Because they were nice for working when the railroad was not still working, and when everything was over, let the camels go. Be free. Now we have 1 million camels that are making a lot of damage. So, if you ignore that you have already been messing around everything, you put a rabbit there, you unleash a positive feedback. But we understand why it happens, right? The vast majority of invaders fail. Some succeed. We know why. And instead, one part of the work we have done is showing that you can actually build what we call ecological firewalls. If you design your microorganisms in such a way that, for example, create not a simple change, but a feedback loop, for example, a kind of symbiotic loop there, you can actually stabilize easily in a very robust way something, and you're not going to spread. It's the ecology what acts as a firewall. But unless you bring the knowledge of ecology into the domain of synthetic biology, there's a lot of really relevant concepts that are still missing there that we can connect.
Great. So unfortunately, we have run out of time. I plan to draw conclusions, but I prefer to have the real expert talk instead. Let me express the fact that, thinking and talking about microbes, this apparently very tiny thing that one could deem as something irrelevant, has brought us to discuss things like, going to Mars or changing capitalism. So I think that is, I just wanted to make this last remark, that really this approach brings in a major change in how we think about things. And so it is clear that there is an opportunity there, and an opportunity that we need to take seriously given the size of the challenges we are facing at the moment. So thanks a lot, Victor, thanks Rachel, thanks Ricardo, thanks Neptune and Monica, and thanks to everybody here. Good night.
Now I can drink because otherwise I'd run off to the bar.
Yeah, your gadget.